Hi, I'm Emma. I'm a bio major at St. Scholastica here, and I just want to share a little bit about my summer research project. So I worked with Dr. Sabrina Scroggins over at UMD this summer, and our research project was based off of preeclampsia, which is a hypertensive disease in pregnancy, and we looked at offspring using mice subjects. And our two main objectives were looking at immune system functions and then cognitive functions in the offspring. So to analyze offspring, we took spleen, thymus, and brain from the offspring after euthanization of postnatal day 8. And to start, we used mini osmotic pumps to induce preeclampsia in a mouse, and then we used a control and we used saline for the control, and then vasopressin was used for the preeclamptic pregnancy. And then once we took the tissues from the mice, we ended up single cell suspension, getting cells down just to a little conical in a tube, and then we analyzed that via flow cytometry to figure out cell populations within those tissues. So some challenges I would say is just beginning to grasp the information. There's a lot of background information with preeclampsia and all of the discoveries that have already been made. And so reading articles was one thing I started out doing just to grasp the information that I was given to understand fully what the research entailed. And so I would say that was a personal struggle of mine. But once I kind of grasped the information, it was very cool to learn the protocols, just research in general, and then a lot of statistical analysis we did at the end to figure out the overall data. So the overall discovery of our research was looking at the brain and glial cell populations within the brain, which are big learning aspects and then just developmental aspects for the brain functioning. And glial cell populations were decreased in a preeclamptic pregnancy versus a non-preeclamptic pregnancy for the offspring. So this can include that difficulties and differences for cognitive function for offspring later in life can occur. And then within the immune system, we also discovered differences within T cells and B cells in the spleen, which can conclude that the cells went through more in a preeclamptic pregnancy for the offspring. So possible autoimmune disease occurrences, and then just more differences within how the cells face pathogens, which was really interesting to me. So immunological differences as well within a normal pregnancy compared to preeclampsia. One of my favorite parts of the research process was just working with my mentors and then my lab mates. It was fun to learn the protocols and then kind of see the results come from that. So I mean, some of the things in lab, it seems pointless until you get to a certain point, and it's just really cool to see the results that came from what we did, and then just the learning processes within the lab were really fun. Now, after concluding my summer research, I'm very excited to go to the conference and just present my findings that we did in lab.